Okay. So, bache, this starts us with our next chapter in general pathology, that is genetics. An extremely nice, simple, straightforward chapter. Let's start with genetics. Starting with extreme basics, that is human genome. Human genome, it has 3.2 billion base pairs. Human genome, it has 3.2 billion base pairs, out of which coding region, out of which coding region means DNA to RNA, RNA to proteins. This is what we mean by coding region. DNA to RNA, RNA to proteins. Coding region is just 1.5%. So I can safely say that non-coding region, I can safely say that non-coding region means DNA to RNA has formed, but RNA to protein synthesis is not taking place. Non-coding region is the remaining 98.5%. Obviously, if coding region is just 1.5%, so non-coding region is the remaining 98.5%. Does this non-coding region have any role in the body? Yes, non-coding region, it plays a major role. It plays a major role in gene regulation. Non-coding region plays a major role in gene regulation. Meaning all the promoters, suppressors, everything is in the non-coding region. RNA तो बना रहा था ना, it is forming RNA. So it plays a major role in gene regulation. That means all the promoters, suppressors, everything is in the non-coding region. So what we have seen till now is human genome has 3.2 billion base pairs. Achha, tell me this here only. What do we mean by a base pair? Base pair means A double bond T, C triple bond G. This is a base pair. Adenine binding with thymine, cytosine with guanine. A double bond T, C triple bond G. There are 3.2 billion base pairs. Coding region is just 1.5%. The remaining 98.5% non-coding region plays a major role in gene regulation. Next point. How many nucleotides form one amino acid? Which is the start codon? AUG. So how many nucleotides? 3 nucleotides. 3 nucleotides form one amino acid. 3 nucleotides form one amino acid. I can divide nucleotides into purines and pyrimidines. I can divide the nucleotides into two categories, that is purines and pyrimidines. Pyrimidine is a longer word. So there are 3 pyrimidines, that is cut. Cytosine, uracil, thymine. There are 3 pyrimidines versus A and G, which are the purines. Okay, basic, you already knew this. So this starts us with our first small topic in genetics, that is point mutations. Which is this starts us with point mutations. 3 nucleotides form one amino acid. जैसे start codon है ना AUG. 3 nucleotides form one amino acid. Mutation in a single nucleotide. Mutation in a single nucleotide is called as point mutation. Out of those 3 nucleotides, if a single nucleotide is mutated, it is called as point mutation. So starting with our first category of point mutations, that is conservative point mutation. पहली वाली से शुरू करते हैं. Let us start with conservative point mutation. Point mutation occurred. A single nucleotide is altered. Point mutation occurred, which leads to formation of new amino acid. Point mutation occurred, which leads to formation of new amino acid, which is biochemically similar, to original. Conservative point mutation. Point mutation hui. A point mutation occurred, which leads to formation of new amino acid, which is biochemically similar to original. Biochemical function conserve हो गया, conservative point mutation. Versus next is non-conservative point mutation. Next is non-conservative point mutation. Non-conservative point mutation means point mutation occurred, but it led to the formation of a new amino acid which is biochemically different. which is biochemically different from original. Non-conservative point mutation. Next is a silent mutation. Next is a silent mutation. Silent mutation means point mutation occurred. A single nucleotide got altered. Point mutation occurred, but no new amino acid is formed. No new amino acid is formed. It is the same original amino acid only. It is the same original amino acid only. This is a silent mutation. So what is the difference between a conservative point mutation and silent mutation? Conservative point mutation occurred leading to new amino acid biochemically similar. Silent mutation में same original amino acid only. Silent mutation. Next is a nonsense mutation. Next is a nonsense mutation. The name tells you it is so nonsensical. Nonsense mutation, it is so nonsensical that it leads to formation of stop codons. Nonsense mutation leads to the formation of stop codons. Which are the stop codons? That is UAA, UAG and UGA. Nonsense mutation. It is so nonsensical that it leads to the formation of stop codons. That is UAA, UAG and UGA. Nonsense mutation. Next point mutation are transitions. Next point mutation are transitions. Transition means purine being replaced by a purine or pyrimidine being replaced by a pyrimidine. Transition, purine being replaced by a purine, that is A being replaced by G or vice versa, C being replaced by T or vice versa. Transitions. Versus transversion. In which purine is replaced by a pyrimidine. Transversion in which purine is replaced by a pyrimidine or vice versa. That is A or G being replaced by C or T. Transversion. Purine being replaced by pyrimidine or vice versa. And the last category of point mutations are frame shift mutations. Last category of point mutations are frame shift mutations. Frame shift mutations, but this occurs because of insertions or deletions. Frame shift mutations. This occurs because of insertions or deletions. What does this mean? Dekho, you very well know that 3 nucleotides form one amino acid. You know this. 3 nucleotides, set of 3 nucleotides form one amino acid. Suppose there is an insertion. A nucleotide is inserted. Insertion okay. There is an insertion. The whole reading frame is shifted. This is called as frame shift mutation. This is a frame shift mutation. All these are point mutations. Mutation in a single nucleotide is called as point mutation. So quickly देख लेते. Let's have a quick recap. कौन सी थी? Conservative point mutation. Point mutation occurred biochemically. New amino acid biochemically similar. Non-conservative. New amino acid biochemically different. Silent mutation. Point mutation occurred, but it is the Same original amino acid only. Silent mutation. Nonsense. It was so nonsensical that it led to stop codons. UAA, UAG, UGA. Transition. Purine being replaced by purine, pyrimidine by pyrimidine. Transversion. Purine by pyrimidine or vice versa. And lastly are frame shift mutations. These are the point mutations. So this is where, this is usually not asked. This is where we actually start with our topic, genetic diseases. Very, very important. It is totally understanding based. Once you just get a hang of it, we'll be very comfortable. We can divide the genetic diseases into 4 main categories. That is Mendelian inheritance, which we can divide genetic diseases into 4 categories. Mendelian inheritance. Mendelian inheritance is of further 4 types. Autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant. ये तो हमें पता ही है. We know this. Mendelian inheritance. Next is non-Mendelian inheritance. Next is non-Mendelian inheritance. Non-Mendelian inheritance, it is of 3 types. That is genomic imprinting. Non-Mendelian inheritance, it is of 3 types. That is genomic imprinting, trinucleotide repeats, genomic imprinting, trinucleotide repeats, and mitochondrial inheritance. Non-Mendelian inheritance, it is of 3 types. Genomic imprinting, trinucleotide repeats, and mitochondrial inheritance. Next, the third category. We'll do all these one by one. Right now, we're just making a table. The third category of genetic diseases are chromosomal abnormalities. The third are the chromosomal abnormalities, which can either be in number or structure of chromosomes. That can either be in number or structure. Chromosomal abnormalities. And lastly is multifactorial inheritance. Lastly is multifactorial. Multiple factors play a role. Whatever we have written on this slide, we'll do it one by one. This is our topic, genetics. So there are 4 categories: Mendelian, non-Mendelian inheritance, genomic imprinting, trinucleotide repeats, and mitochondrial inheritance. Chromosomal abnormalities, which can either be in number or structure of chromosomes, and multifactorial. So let us first start with the easiest of the lot, easiest and a very small 2-minute topic, that is multifactorial inheritance. Let us start with multifactorial. The name tells you everything. Multiple factors play a role. That is more than one genetic plus environmental factors play a role. That is multifactorial. So you tell me, environmental factors matlab lifestyle. The way we keep on having chicken and Pepsi all the time, I'll use the word Pepsi and nothing else, I guess. So multifactorial. Which are multifactorial diseases? Diabetes, hypertension, atherosclerosis associated with lifestyle. Diabetes, hypertension, atherosclerosis. MI, myocardial infarction. MI, congenital heart defects. Cleft lip, cleft palate. Next, acha girls, which is suppose you find a very nice guy. Boyfriend to sabke kami nahi hote hai. I know I should not say that. You find a nice guy, you find your soulmate and you are planning to marry him. Which tablet will you take before pregnancy? Folic acid, to prevent neural tube defects. So neural tube defects, you know that they are prevented by the intake of folic acid. Neural tube defects. Which are the main neural tube defects? That is spina bifida. Neural tube defects. That is spina bifida and anencephaly. All this is multifactorial inheritance. Think of it yourself. If any one of this is given in the MCQs, you will never go wrong in it. Multifactorial, multiple factors played a role along with, that is along more than one genetic plus environmental. So diabetes, hypertension, atherosclerosis, MI, congenital heart defects, cleft lip, cleft palate, and neural tube defects. This was multifactorial. Next, so now we are left with three main categories of genetic diseases. Let us start with our first main understanding based category, non-Mendelian inheritance. And starting with the first non-Mendelian inheritance, that is genomic imprinting. Let us start. But very, very important. Please concentrate. Genomic imprinting. Once you get a hang of the topic, you will never go wrong in it. So starting with genomic imprinting. First point, if you just know that the gene that is getting imprinted, the gene that is getting imprinted, does not express itself. The gene that is imprinted does not express itself. If you know this, you will know everything about genomic imprinting. The gene that is imprinted, जो gene imprint हुआ है, it does not express itself. What does it mean? Genomic imprinting, देखो बच्चे, it is of two types. That is maternal imprinting. We will write everything below this flowchart. Maternal imprinting versus paternal imprinting. It is of two types, maternal versus paternal imprinting. Maternal imprinting matlab gene of the mother is physically present, but it does not express itself. Paternal means gene of the father is physically present, but it does not express itself. What is the name of the disease? With maternal imprinting, it is called as Prader-Willi syndrome. Maternal imprinting is Prader-Willi syndrome versus paternal imprinting, that is Angelman syndrome. Easy to remember. P and P do not go together. So Prader-Willi को paternal imprinting मत बना देना. Prader-Willi is maternal imprinting, Angelman is paternal. And which chromosome are we talking of? Only and only chromosome 15q. P for petite. Petite is the short arm. Q is the long arm. We are only talking of long arm of chromosome 15. Normally, we get one chromosome 15 from the mother, one from the father. This is normal. All of you very well know this, कि we are diploid, we are 2n. We get 23 chromosomes from mother, 23 from father. You know this. In Prader-Willi syndrome, chromosome of the mother is physically present, but it does not express itself. Maternal imprinting. Plus, what is happening to the chromosome of the father? Paternal deletion. This is Prader-Willi. बच्चे कुछ भी नहीं है. If we have to do something scandalous at home, mother and father both should not be there. Mother is there but she is sleeping. Maternal imprinting. Plus father is not at home. Paternal deletion. This is Prader-Willi. So you tell me what is Angelman? It is paternal imprinting plus maternal deletion. This is Angelman. We made the mnemonic P and P do not go together with imprinting. Question को ध्यान से देखना. See the question carefully. Paternal deletion is Prader-Willi. Paternal imprinting is Angelman, firstly. So imprinting plus deletion. This is the first method of having the disease, accounting for 70% cases. This is the first method. Continue the same flowchart. The second method of having the disease is disomy. Disomy means both chromosomes come from same parent. Disomy means both the chromosomes come from same parent. Which chromosome are we talking of? Only and only chromosome 15. हम सिर्फ chromosome 15 की बात कर रहे हैं. So you tell me, Prader-Willi is which disomy? I want all of you to commit to the answer. Prader-Willi, both the chromosomes came from the mother. It is maternal disomy. They came from the mother, but they do not express themselves. Remember, Prader-Willi was maternal imprinting now? It is maternal disomy. Versus Angelman syndrome, which is paternal disomy. Versus Angelman, which is paternal disomy. You cannot ratify this, you have to understand it. अगर हमें पता है कि the gene that is getting imprinted does not express itself, we know everything about it. तो कोई scandalous करना है तो दोनों parents नहीं होने चाहिए. Prader-Willi में mother is there but she is sleeping, father is not at home. Maternal imprinting plus paternal deletion. Angelman उल्टा है. Second method था disomy. Lastly, clinically, continuing the same flow chart. Clinically, if mother is sleeping, we sit in front of TV and sit in front of TV and eat food whole day. मम्मी junk food नहीं खाने देती ना. So it is associated with hyperphagia. We are not going out to play basketball. Hypotonia, short stature. Hyperphagia, hypotonia, short stature. Hypogonadism, and mental retardation. Hypogonadism and mental retardation. Prader-Willi. Mother is sleeping, so we sit in front of TV and eat food whole day. Versus Angelman syndrome, if father is sleeping, girls, बच्चे तब हम खाने की नहीं सोच रहे. We are thinking कि चलो let's talk on phone happily, नहीं तो पापा उठ जाएंगे. So Angelman syndrome, the classical term that is used is happy puppets. The classical term is happy puppets. That is uncontrollable laughter. That is uncontrollable laughter with ataxic seizures. Uncontrollable laughter with ataxic seizures. And this is also associated with mental retardation. This is genomic imprinting. कुछ भी नहीं था. Just remember it like a story and you will not go wrong in it at all. This is genomic imprinting. The gene that was getting imprinted does not express itself. Which chromosome were we talking of? Only and only chromosome 15q. Long arm of chromosome 15, nothing other than that. This was genomic imprinting. Next point. But this gets us to our next non-Mendelian inheritance. Which is the next non-Mendelian inheritance? That is trinucleotide repeat diseases. This gets us to trinucleotide repeat diseases. The classical trinucleotide repeat disease being Fragile X syndrome. Remember, there are 3 non-Mendelian inheritance now. एक बार दोबारा देखो. Which were the 3 non-Mendelian inheritances? This was the category of genetic diseases. Mendelian, non-Mendelian. We are done with genomic imprinting. Next non-Mendelian inheritance is trinucleotide repeat diseases. Classical trinucleotide repeat being Fragile X syndrome. And last one is mitochondrial inheritance. So in this section we will cover non-Mendelian inheritance. So the second is trinucleotide repeat diseases, the classical being Fragile X syndrome. First point. Which is the most common genetic cause of mental retardation? Most common genetic cause of mental retardation. Answer बच्चे, Down syndrome. Most common genetic cause of mental retardation is Down syndrome, followed by Fragile X syndrome. Downs, followed by Fragile X syndrome. So I can safely say, second most common genetic cause of mental retardation is Fragile X. ठीक है, fair enough. Fragile X syndrome, it is associated with FMR1 gene mutations. Fragile X, FMR1 gene mutation, that is familial mental retardation 1. It is associated with FMR1 gene mutation. Familial mental retardation 1 gene mutation. FMR1 gene mutation present on X chromosome. It is a trinucleotide repeat disease. Which trinucleotides are getting repeated? The trinucleotides getting repeated are CGG. देखो, easy to remember. For the Hindi speaking, patient is mentally retarded, तो patient को कम ज्ञान है, patient को कम ज्ञान है. Trinucleotides are CGG. For the English, I mean, don't worry about it, you just have to remember it. Trinucleotides getting repeated are CGG. What does this mean? देखो, normally, all of us, we have 30 to 60 repeats. मतलब CGG, 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 इस तरीके से it is 30 to 60 times. Then there is a pre-mutated state. There is a pre-mutated state where there are 300 to 400 repeats. And lastly is a mutated state where there are 3000 to 4000 repeats. Number of repeats is increasing in multiples of 10. Does not matter. What is very important is the mutation increases at oogenesis, which is very, very important. The mutation increases from mother to the child. Meaning if mother is in pre-mutated state, her child will be in mutated state. So who will be more severely affected? Mother or the child? The child. This principle is called as anticipation. We are anticipating that the child is more severely affected than the mother. This is called as anticipation or Sherman's paradox. It is paradoxical. Parents are supposed to protect us, na? Here the disease is increasing from mother to the child. So this is called as anticipation or Sherman's paradox. That is increasing severity of disease. That is increasing severity of disease with successive generations. The severity of the disease is increasing from mother to the child. Increasing severity of disease with successive generations. Called as anticipation or Sherman's paradox. In Fragile X syndrome, which trinucleotides were getting repeated? CGG. Where were they getting repeated? In non-coding region. CGG. They are getting repeated in non-coding region. This is our first trinucleotide repeat disease, Fragile X syndrome. Put a line on the side. Another trinucleotide repeat disease. Is Huntington's chorea. Which is easy to remember. पहले मुझे ये बताओ, first tell me what is chorea? Purposeless involuntary movement. It is purposeless and involuntary. That is chorea. Huntington's chorea. Hunter keeps the animal in cage. CAG. These trinucleotides are getting repeated in coding region. They are getting repeated in coding region. And the trinucleotides which are repeated in coding region, in them, the mutation increases at spermatogenesis. That is from father to the child. Now it does not matter whether the mutation increases at oogenesis. देखो, जो non-coding region repeat diseases थी, in them the mutation increased at oogenesis. Versus in coding region, the mutation increased at spermatogenesis. Does not matter. Both of them show anticipation or Sherman's paradox. That is increasing severity of the disease with successive generations. This is Fragile X syndrome. Trinucleotide repeat disease. Which trinucleotides were getting repeated? CGG in Fragile X, CAG in Huntington's chorea. And lastly, clinical features associated. Clinical features, Fragile X syndrome. Very obvious. It was the second most common genetic cause of mental retardation. So it is associated with mental retardation, obviously. Next point in Fragile X, which everything is large. That is, it is associated with large forehead, large jaw. What is large jaw called as? Macrognathia. Large jaw, large testis, that is macroorchidism. Large testis and large ears. So everything is large. Large forehead, large jaw, large testis, large ears. Fair enough. Which is the most characteristic feature of Fragile X? These are your options. Can I say mental retardation? No. It was not even the most common. It was the second most common genetic cause of mental retardation. So most characteristic feature, macroorchidism. Most characteristic, macroorchidism, which is detected at puberty. Macroorchidism, which is detected at puberty. Next point. Which part of the face is not large? Which part of the face is not large? Just touch your nose once and you will forever remember which पूछा जाता है. Thankfully it was the nose. What is not large? Nose. First class. So this finishes our second non-Mendelian inheritance, trinucleotide repeat diseases. And this gets us to our last and the smallest category of all, that is mitochondrial inheritance. This gets us to mitochondrial inheritance. सबसे पहले, first of all, tell me this. We get mitochondria from mother, father या both? Only mother. We get mitochondria only from the mother. So it is maternal inheritance. Mitochondrial inheritance, we get mitochondria from the mother. It is maternal in nature. It is maternal inheritance. So it has a simple pedigree chart. If the mother is affected, all the children are affected. If mother is affected, all the children are affected. Asha, I need not tell you now कि circle is female. देखो girls गोल गोल बातें करती है ना, circle is female. Square, we guys, we are simple minded. हमें ज़्यादा बातें करने नहीं आती. Square is male. You know this. So if mother is affected, all the children are affected. If father is affected, none of the children are diseased. Plain and simple. This is the pedigree chart. This is the pedigree chart of mitochondrial inheritance. It is maternal inheritance. Next point. So I can safely say DNA is of two types. That is nuclear DNA. DNA is of two types. That is nuclear and mitochondrial DNA. Which has higher risk of mutations? Nuclear DNA or mitochondrial DNA? Which has more mutations? Mitochondrial DNA. Mitochondrial DNA is at increased risk of mutations. It is at increased risk of mutations. Why? Because DNA repair genes are absent. In mitochondrial DNA, DNA repair genes are absent. If a mutation has occurred, it will persist. DNA repair genes are absent. And secondly, it is closer to oxidative phosphorylation. Remember, oxidative phosphorylation takes place in mitochondria, leading to synthesis of free radicals. It is closer to oxidative phosphorylation. Closer to which free radical? Hydroxyl free radical. ठीक है, fair enough. This is not even asked. So mitochondrial DNA has increased risk of mutations. Does not matter. There are only 2 points that you need to know with mitochondrial inheritance. The first was the pedigree chart. You need to know the pedigree chart. And the second point is diseases associated. Bache has to be remembered. Just see this table once or twice and you will be very comfortable. Throughout your 5.5 years of BDS, you will never do them anywhere else except in this list. देखो खुद सोचो, if mitochondria is not working, Role of mitochondria is synthesis of ATP. ATP synthesis reduces, leading to lactic acidosis, encephalopathy, and stroke-like symptoms. Stroke क्यों हुआ? Stroke is reduced blood supply to the brain. Here, blood supply to brain is okay, but the patient has stroke-like symptoms. Blood is reaching the brain, but it is not getting utilized. Mitochondria is not working. Muscles are getting affected. Which muscles? The ones which continuously work, that is muscles of the eye. So diseases. Most common disease with mitochondrial inheritance is MELAS. That is mitochondrial encephalopathy. Most common disease is MELAS. Mitochondrial encephalopathy. Lactic acidosis, and stroke-like symptoms. Most common disease with mitochondrial inheritance is MELAS. Mitochondrial encephalopathy, lactic acidosis, stroke-like symptoms, MELAS. Other diseases are Pearson's syndrome. I'm just writing the full form Atma ki santushti ke liye. You will never be asked. P for Pearson's, P for pancreatic insufficiency. Pancytopenia. ATP are not getting used. So bone marrow, the cells are not multiplying. Pancytopenia and lactic acidosis. No need. Just Pearson's syndrome. Simple question. Most common mitochondrial inheritance, MELAS. Pearson's syndrome is autosomal recessive, autosomal dominant, X-linked recessive, mitochondrial. Other diseases with mitochondrial inheritance are KSS, Keene-Shirey syndrome. Keene-Shirey syndrome. LHON, Leber's hereditary optic neuropathy. See, if you just see this table once or twice, you will be very much okay with it. LHON, Leber's hereditary optic neuropathy. LHON. And MRF. That is myoclonic epilepsy. Ragged red fibers. These are the diseases associated with mitochondrial inheritance. Just speak them out once with me. Most common was MELAS. Mitochondrial encephalopathy, lactic acidosis, stroke-like symptoms, MELAS. Others are Pearson's syndrome, pancreatic insufficiency, pancytopenia, lactic acidosis. KSS, Keene-Shirey syndrome. LHON, Leber's hereditary optic neuropathy, LHON, and MRF, myoclonic epilepsy, ragged red fibers. So, bache, this finishes non-Mendelian inheritance. It was of 3 types: genomic imprinting, trinucleotide repeat diseases, and mitochondrial inheritance. Done.